Hello and welcome to the second part of our Year 12 Biology non-Mendelian inheritance patterns. Last week we focused on incomplete dominance, codominance and sex-linked inheritance. If you're not 100% on them, feel free to pause the video and go back and watch the first one. This week we're focusing on polygenic inheritance, otherwise known as multiple alleles, lethal alleles and pleiotropy. Your key syllabus objectives today are to define polygenic inheritance and predict frequencies of genotypes and phenotypes using different alleles. Make sure you are really familiar with the definitions of these two terms. So here is your definition of polygenic inheritance. Make sure you write it down. Polygenic inheritance is a form of inheritance where a single phenotype is determined by the combined effect of two or more different alleles or genes. For instance, skin color. In humans, we actually have 3 different genes that codes for our skin color. Other names for this that you might see in your research is multifactorial inheritance or quantitative inheritance. The production of melanin, which is a pigment that determines our skin color, is controlled by 3 genes. The amount of pigment in a person's skin is directly proportional to the number of dominant genes the person inherits. So if we say that there are 3 genes, gene A, gene B, and gene C, this person here with the lightest possible combination of skin color has inherited the homozygous form of each of the 3 genes. And this person here, this girl here, has inherited the homozygous dominant form of all 3 genes. And people in the middle here have intermediate forms. So this person may be big A little a, big B little b, big C little c, heterozygous for all 3 genes. We end up with a continuum from very pale all the way through to completely black skin. And we've divided up into 7 different shades of skin color, ranging from really, really dark, the higher the color, the higher the number, the more melanin you produce, all the way through to very pale, where you've got the homozygous recessive of all 3 different alleles, 3 genes. So when we're predicting the gamete production, if we have a heterozygous intermediate skin-colored person here, they've inherited 1 dominant and 1 recessive for each of the 3 genes. Let's say this is a male person. They're going to produce sperm. And into each of those sperm will go 1 allele for each gene. So it could be the dominant form or it could be the recessive form. So there are 8 possible different combinations or different sperm types that could be made with this gene or this phenotype. So you could have the homozygous dominant, you could have the homozygous recessive, you can have all the different combinations. And there are 8 different forms that it could take, all the way from homozygous dominant to homozygous recessive. So if we're going to cross 2 people of intermediate skin color who are heterozygous for each of the 3 genes, we're going to have, I'm just going to get rid of these ones for a second. We're going to have all the different possible sperm and eggs or gametes listed up here and down here for the 2 parents. From there, you can make the Punnett square, which is going to be enormous. There's going to be 64 possible offspring here. And this one, you're going to get the homozygous dominant form. And there's only 1 possible way that you could get that in this whole Punnett square. So the chances of getting an incredibly dark-skinned person or offspring is 1 in 64. And there's only one way to get the homozygous recessive in this Punnett square as well. So they have a 1 in 64 chance of having this genotype or this phenotype. Pause the video here. I'm going to hand you out one of these tables and you're going to have a go at doing all the predictions or the predicted genotypes and phenotypes. So do the genotypes by having a look and going, okay, you've got big A, big B, and you've got heterozygous for C, big C, little c. This one, you'd have big A, one from mum, one from dad. You'd have heterozygous for B, big B, little b, and you'd have dominant from both parents for C. You've really got to watch what you're doing as you're doing this. Big A, big B, little b, heterozygous, and big C, little c, heterozygous for C. Pause the video now and have a go yourself on your paper. If you're doing this from home, screenshot this and have a go at doing it either on a Word document with your stylus or doing it the long way. You can create your own table and do it in Word. I would probably recommend printing this slide and just doing it on paper because it's a whole lot easier. When you've finished, grab a highlighter and highlight which ones are going to be extremely dark, which ones are going to be extremely light, and see if you can find a pattern in amongst them. Pause the video here. Okay, hopefully you've had a chance to do that. And you should find that you get something like this. As we found before, the homozygous dominant, you've only got a 1 in 64 way of getting that. And the homozygous recessive, you've got a 1 in 64 chance of that as well. But there are different ways to get the different combinations. Big A, heterozygous for B, heterozygous for C. There are different ways to get those combinations. So you end up with a one way of getting each of the homozygous, but there are 6 ways of getting the two either quite dark or quite light. There are 15 ways of getting the next one, and there are 20 ways of getting the completely heterozygous. Big A, big B, little b, big C, little c. So there are 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1 is your phenotype and genotype ratio for this. And this correlates to the different numbers that we were talking about. There are 6 different shades all the way from 0 to 6. 0 being our lightest skin color, all the way through to 6 representing the darkest possible skin color in humans. And you'll be able to recognize the different colors skin with our different genotypes. So this is actually a phenotype table representing skin colors. And when we graph that, we find that it fits a beautiful bell curve. Oops, bell curve. Not sure what I wrote there, but it should say bell curve. Bell curve. So we have this lovely bell shape with our graph, where very few people have the darkest skin color, and very few people have the lightest skin color. Most people have an intermediate form, and we are within 1 or 2 standard deviations from the mean skin color. And you find that that happens over and over again for the different skin types, the different colors, and the different genotype and phenotype combinations. And we call this continuous variation because skin color is a continuum. You can be everywhere from the absolute lightest to the absolute darkest, and people fall somewhere in the middle along this bell curve. Skin color isn't the only continuum that we find, and it's also a polygenic trait. Polygenic traits generally form this beautiful bell curve because you end up with this continuous variation. Human height is also one of them, where most people fall somewhere around the mean, and very few people are at the tall end, and very few people are at the extremely short end. Okay, on to the next form of of non-Mendelian inheritance. And we want to talk about lethal alleles here. Some combinations of alleles are deadly. They're lethal. They kill the offspring. Usually they're lethal in their homozygous combination. Our example is Manx cats. Manx cats have no tails. Their owners haven't lopped them off in some barbaric act. These cats are born with no tail, and it's coded for by a dominant allele. So we represent that as big M with a little L for Manx. And the recessive form codes for a normal tailed cat. So our possible genotypes then, you could have homozygous dominant, big M, big M. This is lethal. The cat dies before it's even born. So you never get to see this homozygous dominant form. All Manx cats are heterozygous. So if it has no tail, it is a heterozygote. And the homozygous recessive form, it has a tail. It's a normal cat. So let's do some prediction. This is what we're talking about when we say to predict something. We want you to do this combination. So 2 Manx cats are crossed. So we know they have to be heterozygous. So the father's genes, we're going to have big M, little m, big M for mum, little m for mum. So when we get the big M, this is lethal. None of those cats are going to be born. This next offspring is going to be big M, little m, so it's a heterozygote, it's a Manx cat. This one's going to be a heterozygous, a Manx cat, and then we've got 1 little m and 1 little m from mum and dad, so this one is a homozygous recessive. Okay, what did our question ask us to do? What are the genotype and phenotype ratios? Okay, genotype. Well, we only have 2 genotypes being born here. The heterozygous form and the homozygous recessive form. And you get 2 heterozygous to 1 homozygous recessive. So that's your genotype ratio. Your phenotype ratio, we've only got 2 phenotypes here, Manx and normal. It's really important that you define your ratios so that we know which one is which. So Manx, we've got 2 of them, and normal, we've got 1. So here are your genotype and phenotype ratios for this prediction. Pleiotropy. In pleiotropy, a single gene can sometimes have effects on different traits. So we're going to give you 2 examples here, sickle cell disease, which we're going to do in this video, and the disease PKU, which you're going to find in your textbook. I want you to go and read about that. So in sickle cell disease, not only does it form malformed or sickle-shaped red blood cells, but having this gene, this beta-globulin gene mutation, also affects blindness, can cause blindness, it can cause liver failure, and it can cause heart attack. So one gene, very different systems, very different phenotypes. So that's pleiotropy. One gene, many different traits being caused by the one gene. The last one we're going to talk about today is epistasis, because I find that a lot of students mix up pleiotropy with epistasis. Remember, pleiotropy, one gene, lots of different effects. Epistasis, however, is when one gene can cancel the other. So if I've got gene B and it can actually switch off gene A, then we say that gene B is epistatic to gene A. An example of that is albinism. An animal that inherits 2 recessive alleles for the gene for albino is going to end up with white fur. But somewhere else on its chromosome, up here and down here, it'll have a gene for albinism and it'll have a gene for colour of fur or skin. Having this recessive albino combination means that this other gene gets switched off. So regardless of whether it has a black, a blue, a red, or a hot pink gene for fur colour, if it's got this albino combination of alleles for this gene's going to be switched off and no colour is going to come out in the fur. So that's epistasis in action. And that can happen in all different animals. Things that fly, things that live in the water, humans, monkeys, apes, orangutans, koalas, you name it, there are albino versions of it. Another example of epistasis in action is male pattern baldness. Having a gene for baldness means that regardless of what your gene for hair colour says, you're going to end up being bald. Because this gene switches off this gene. Okay, that is all I wanted to go through with you this week on non-Mendelian inheritance. So we have now finished incomplete, codominant, sex-linked, polygenic inheritance, lethal alleles, and pleiotropy. And we threw in epistasis for good measure, just to make sure that you don't mix them up. We are now done with non-Mendelian inheritance. What I want you to do now is to go to our class OneNote and have a look at the activities that we have for you today. If you're at school, your teacher will have print versions of the worksheets available for you. If you are at home, you're going to either need to do them digitally on your device or you're going to need to print them out. Thank you very much for listening.